Professor James Zhou is an associate professor at Stanford University. He mainly is working on foundation of large language machine learning and in-depth scientific and clinical applications. Many of his innovation has been widely used in tech and biotech industry. He got lots of awards, including the prestige Sloan Fellowship, Overton Prize NSF Career Award, 2 Chan Zuckerberg Investigator Award, a Top 10 Clinical Achievement Award, several Best Paper Awards, and a faculty award from Google, Amazon, Adobe, and Apple. And his research has also been profiled in popular press, including New York Times, Wall Street Journal, and Wired. Let's welcome Professor Zhou. Okay.
Great. Thank you very much for the introduction and for organizing this very timely and interesting workshop. Yes, I'm excited to participate, and I want to share some of the recent directions we're in exploring around building AI scientist agents. In particular, these agents that can help us with biomedical discoveries. So, usually when we think about using AI in biomedicine, right, in science, right, AI is often thought of as a tool for solving some well-defined and often pretty narrowly defined problem, right? So, for example, if we have the problem of predicting protein structures, right, then tools like AlphaFold are very useful for predicting those structures. Or if I want to diagnose and maybe analyze these kind of ultrasound videos, then we in the past have built these tools like IcoNet that can help us to segment these videos and assess the different heart diseases from these ultrasound videos. But in each of these cases, right, so there's actually a lot of work that goes into figuring out what data should be go into these tools, and also how to interpret the outcomes, right, the results from these tools, right? That's really requires a lot of human work. And what I think is quite interesting over the last couple of years is really this paradigm shift, right? Where now we can potentially have AI models powered by advances in large language models that can be much more flexible, right? So in addition to just solving these narrowly defined problems, these AI models can also come up with research plans, right, hypotheses, and data analysis. They can basically do a lot more things. And in particular, these AI agents, right, by leveraging these language models under the hood, they themselves use different tools like AlphaFold, but they can also communicate with other agents and also with other researchers, right? So this potentially opens the door of having these AI not as a tool, but as more of a teammate across the research and development. So that's the part that I'm very excited to explore. And I want to share a couple of examples of how we built these AI scientist agents. So in the first half, I'll describe the virtual lab. So the virtual lab is a platform that we build where you can have different AI scientist agents with interdisciplinary expertise, right, that work together to tackle challenging open-ended research problems. So in the virtual lab, there's an AI professor, so also called an AI principal investigator. And under this AI professor, there's actually a bunch of different AI students. You can think of them as having different expertise, right? In, let's say, in immunology, data science, computational biology. And they work together with the AI PI and also with the human researcher to tackle these problems. And we also developed a mechanism that we call the virtual lab school, whereby these AI agents, right, they could actually learn to develop deeper expertise into specific scientific domains. Right, so I'll also explain a bit of how the virtual lab school works. And this work is led by a fantastic PhD student at Stanford working in my group, so Kyle Swanson. So here's the overall design of the virtual lab, right? So when when myself, let's say, interact with the virtual lab, we mostly can do that by talking to the AI PI. So for example, I can tell the AI PI, I'm interested in analyzing this kinds of data, or I'm interested in designing this kinds of binders, right? Or doing this kind of biomarker discoveries. I can just provide the high-level problem description to the PI agent. And then it's the job of the PI agent then to automatically to create and then to train the sub-agents that have the relevant domain expertise, right? So maybe for a particular problem, the PI agent might decide it's useful to have an immunologist on the team, right? And then the PI would actually create this immunologist where maybe it's useful to have a computational biologist on the team, right? Then the PI would actually create this computational biologist. Right, so it's actually a job of the PI agent then to identify what areas of expertise are needed, and then to create the agents that have those relevant expertise. So behind the hood of each of these agents could be a large language model, right? So we have used GPT-4o, but we've also used these reasoning models. And I'll actually come back to in a few slides of how we can take the existing language models, right, but then further fine-tune them to have the agents that have deeper expertise in these specific scientific domains. Once the agents are created, then they can have group meetings where all the agents meet together to discuss a research topic. They can also have individual meetings where one of the agents actually meet with the critic or the professor agent, right, to try to solve a specific subtask. So here's a little bit more detail about how these group meetings work. Right. So in the virtual lab team meeting, basically what happens is that the PI agent will set up, here's the meeting agenda. Maybe there's some questions, specific questions that you want to answer by the end of this meeting. Then all of the other agents, right, they will all contribute sequentially towards answering those set of questions by providing expertise from their own perspective. Right. So to make it more concrete, so here's an example of a virtual lab team meeting, one of the actually earlier meetings. So here, basically, we set the scientist agents the goal of developing and designing new binders to the recent COVID variants. But we don't tell the agents how they should do it or what they should try to design. So then the professor agent, the PI, basically said, okay, so our goal is to develop some effective binders to the recent spike protein, right, of the recent SARS-CoV-2 variants. In this case, the immunologist agent spoke up next and made the interesting recommendation of actually try to design nanobodies. So this is already kind of an interesting decision because usually when we think about designing binders, right, most of the we think about designing antibodies, right, which are more available and more common. And nanobodies are basically sort of smaller cousins of antibodies, right? Whereas for antibodies, you can find them in many animals. Nanobodies are only found in a few animals like camels. Right, so it's kind of already a surprising decision made by the immunologist, but as we'll see in a bit, right, this actually turned out to be quite a wise decision. So here the machine learning specialist actually said, okay, I agree with this focus on antibodies because they are smaller, right? So that actually makes my job easier, right? Because I can then develop deep learning models to predict their structures. The computational biologists provide a little bit different perspective that says that, okay, so in addition to using these deep learning models, let's also use these more physics style, like molecular docking and simulations, right, to make the modeling more robust. In all of these meetings, we also have a critic agent, right? So this is sort of like a reviewer that sits in on every discussion, right? So this critic agent is meant to be more skeptical and provide more skeptical feedback. Right. In this case, the critic agent says that, okay, well, we don't have a lot of public data, right, around nanobodies, right, for COVID, so we need to be especially careful around overfitting. So the actual meeting go on for longer, right? They can have multiple rounds of discussions, but here I'm just showing you sort of the verbatim part of the first part of the meeting to give you a sense of how the agents can come up with different ideas, right? Here they come up with like a new research plan of designing, optimizing nanobodies using deep learning and also using these molecular simulation tools. So the goal of these group meetings is for the agents to basically come up and to decide on the high-level research plans, right? Once those plans are decided, then the professor agent will assign specific tasks to each of these agents to solve, right? And then they would do that through one-on-one meetings where each of the agent then would actually solve a particular task. Like maybe one of the agents would try to write a piece of code, then the professor will meet with that agent to debug and to review the code. So one of the nice things about these AI scientists here is that their meetings are more efficient than our human meetings. Right? So one of their meetings maybe would take a few seconds, maybe a minute. So in the time that we had our coffee breaks, they already had maybe hundreds of meetings. And also because these AI scientists, they don't get tired, they actually run multiple meetings in parallel. Right? So basically for every decision that's made by the virtual lab, we run 5 meetings in parallel. In each of those parallel meetings would discuss, they would discuss the same questions, but they will often come up with different ideas, different suggestions across these different parallel meetings. So it's actually quite an effective way to explore a broader range of potential ideas. And then it's the job of the professor agent to read the transcripts from all these parallel meetings and then to make a consensus decision, right, of what to do next. So in case you're curious, here I'm also showing in this pie chart basically how often the different participants would speak in these virtual lab meetings. So we, the human researchers, like myself, my students, we would speak not very much, about 1% of the time. So our job is to mostly provide some high-level directions, right? We tell the agents we're interested in designing binders to COVID. We can also tell the agent some of the constraints we have. For example, here's how much budget we have to do the physical experiments. And we don't really want to micromanage these AI scientists, right? So we really want them to do their own designs and be creative and do their own work. So the PI agent also speaks quite a bit, about 20% of the time. In this case, the computational biologist agent is the person that spoke the most, because this agent has the most work to do, right? Because the goal here is to design these nanobodies, then the computational biologist has to then do a lot of the work in coming up with those pipelines. You can see that the scientific critic agent also speaks quite a bit, right? It actually provides feedback to every decision made by all of the other agents. The machine learning specialist here is particularly useful for writing all the code, right? So for example, all the code in the virtual lab are all written by the machine learning specialist, right? So that agent sort of also plays a critical role. So then through this process of these group discussions and then one-on-one work meetings, right? So then the virtual lab starts to make rapid progress towards this goal of designing binders for the recent COVID variants. And here the agents actually did something I think is quite interesting, right? They decided to come up with like a new workflow of optimizing these little proteins, these nanobody proteins, right? So the agent decided they want to actually take some of these existing tools like ESM, which is the protein language model, and AlphaFold and Rosetta, but they would also modify these tools and then put them together into this new workflow, which is different from what we've seen before in previous publications. So essentially what this virtual lab workflow, design workflow does is that first the agents would use this protein language model called ESM to filter for proteins that are likely to be sort of stable nanobodies. Right, so ESM would just actually take a input, this amino acid sequence, and computes an embedding and also a score that quantifies how likely is that sequence to form like a structurally stable protein. Right, so the agents would use that to filter for some initial candidates, and then they will put each of those candidates into AlphaFold together with the spike protein of the target SARS-CoV-2 variant. Right, and the AlphaFold will generate this three-dimensional structure of how the candidate nanobody potentially would bind to this target spike protein. Then the agents decided to take this AlphaFold three-dimensional structure as the input into Rosetta, which would do more like a physics style, more like a relaxation of that structure, and then molecular simulations to see how what will be the binding affinity between the nanobody and the target spike protein. Right. So through that process, then the agents actually gets like a good estimate, a good sense of how promising is this nanobody candidate, and they would actually run this whole loop multiple times to sequentially filter for the most promising candidates. So I think this is actually quite an interesting workflow. And other kind of part that's interesting is that the agents also made some modifications to these tools, right? For example, with this protein language model ESM, the previous papers, publications, had a particular method of computing scores from ESM, right? But the agents decided to do it in a different way. But in retrospect, I think the way that agents did it also makes quite a bit of sense. So all of this computational pipeline is designed and implemented by the AI scientist agents, right? And they run this pipeline to come up with some of these candidate nanobodies. And where the human collaboration is very useful is that we want to take these nanobodies designed by the AI scientists and then actually make them in the real lab, right? And then see and test in the real world how well do they work. This is where we collaborated with my collaborators, John Pack's group at the Transact Work BioHub, where here the agents would actually, so we took basically several of these nanobodies designed by the agents. So here I'm showing you 2 example nanobodies. And we synthesized them. And then we basically performed the standard ELISA experiments to measure how well do these nanobodies actually bind to the spike proteins of different COVID variants. So that's basically shown here in these two bottom plots. So let me parse the different curves here. So if we first look at this bottom curve with the open red circles, this basically measures if we take the existing nanobodies designed by humans, like how well does it bind to these recent COVID variants called the JN1 variant, which started to spread last year, right? You can see that it does not show really binding to this JN1 variant. And then the solid red curve is basically after the AI scientists had modified the existing nanobody by introducing some of these mutations, now you can actually see more promising binding, right, to this JN1 variant. And these other two curves basically also shows if we take the same nanobodies, right, and how well does it actually bind to a very different variant of COVID from Wuhan, right, from 5 years ago. So this is the, you can see that basically after the nanobodies designed by the AI scientists actually shows even enhanced binding, right, to the original Wuhan strain of the virus. And the reason why it's quite interesting and exciting is that this now shows signals of the same nanobody, right, designed by the agents, showing promising binding across these very diverse virus, right, from the virus, COVID virus from Wuhan, and the virus from last year, JN1, actually had undergone many, many mutations, right? But despite those differences, the nanobody designed by the agents actually seems to show pretty consistent binding, which is ideally one of the properties that you want to have in any potential a potentially good vaccine. And of course, I want to caution that we still need to do more experiments to validate these nanobodies designed by the agents. But this is already showing quite promising signals that the agent can take a very challenging problem that we have not been able to solve, right? And then design their own computational pipeline, implement those pipeline, and then really come up with new nanobodies that we can experimentally validate in the real world. And this is all done relatively quickly. So I want to then come back to a little bit more about this aspects of how do we train these agents, right? Because we can initially use the sort of off-the-shelf language models like R1 or GPT-4 or 4.0 or O1 mini, right? But those models often run into challenges when they're thinking about these deep technical domains that requires more knowledge or more recent information. Right, so to mitigate this, we created what we call a virtual lab school, basically where AI scientists can actually go to our school and then design their own learning curriculum to teach themselves on how to become a deeper expert into one of these domains. So how the virtual lab school works is that we would tell the agents, right, so here are some of the topics that you should learn about. Maybe you should learn about the recent COVID variants or learn about using AlphaFold. And then the agents will actually take each topic and come up with a set of learning questions that are relevant for that topic. And then based on that, it will go out, we equip it with different search engines, right? So the agents can go out and try to retrieve information, for example, from all the open access papers. It will filter for papers that are the most relevant, and then do a deep read of the entire paper, right, including the methods and supplements. As it's doing those deep reads, right, it would actually then start to fine-tune the model parameters as it's ingesting more information. So that even though initially they're sort of being maybe perhaps the same agent, right, the same base language model, but as the different agents learn on different domains of expertise, then the model starts to diverge because their parameters also starts to diverge. So after each scientist agent, does its learning, right, it will actually do a self-assessment to see how well did it actually learn that particular topic. If it did not learn it very well enough, it could actually go back to the virtual lab school and do additional training. This process is fairly autonomous, right? Basically the agents come up with their own learning curriculum, they find the relevant information, and then they would update their own parameters and teach themselves on how to be an expert into those domains. So our goal here then is to, through this virtual lab school, right, we essentially want to create a broad roster, right, of hundreds of different AI scientists, each equipped with a deep expertise in one of these domains. Right? And then these different scientists can work together, right? So across the different kinds of research problems that one might encounter, from earlier stages of maybe biology and drug discovery to later stages of clinical trials, right? These different AI scientists can actually work together with each other and also with human teams to help to speed up and to augment the research R&D process. So one of the challenges that we have, broadly speaking, was designing these more complex multi-agent workflows, right? Is how do we try to really optimize these agents? And I think this is also still an ongoing challenge in the field, an open problem. And we recently took some steps to try to address that problem by developing a system called TextGrad as a way to basically try to optimize these agentic workflows. So the motivation for this is that if I have like a standard deep learning model, like a neural network, then it's relatively easy to optimize it just by using the backpropagation of numerical gradients. Right? So that's basically enabled all the advances in deep learning over the last 10, 15 years. But this backpropagation for numerical gradients for automatic differentiation does not really work in the context of agents, because the agents themselves could be, let's say, maybe you only have API access to an agent, right? So you can't really differentiate through it. Or maybe the agents use different tools, which are like physics simulators or search engines. You cannot differentiate through those. Also, the agents can use different, they talk to each other through human language, which are also not differentiable. Right, so that makes optimization of these agents not feasible using a standard differentiable optimization. To mitigate that challenge, we created a system of non-differentiable optimization that we call TextGrad. The idea here is to basically replace these numerical gradients with what we call text gradients, right? So basically each text gradient is a piece of text feedback that locally tells one of these agents or one of these components how to locally improve its behavior or improve its performance. So here's an example of what the text gradient will look like. So let's say if I have a task of designing and optimizing these new molecules, right? So the agent's trying to optimize these new molecules. So then each of these text gradient now will be a piece of instruction, right, to the agent on how to improve this molecule, right? So in this case, maybe the first gradient would say you want to add more of these hydrogen bonds, right? And then the agent might implement that by adding these more, by modifying the molecule. And then the next gradient would say, okay, maybe you want to add like a aromatic ring to enhance this binding, right? And the agent will follow that gradient basically by following that instruction, right? So then you can see that each gradient now is basically a piece of instruction to the agent on how to locally modify its current design of the molecule, right, to enhance its property. And through experiments, we've shown that this text-based optimization, right, is actually quite efficient. You can start with a very generic molecule, and after just less than 10 iterations, convert into a more complex molecule that actually has quite good biophysical properties and binds well to these important drug targets. And we've also shown through experiments, right, that one of the benefits of these text-based optimization is that they cannot be converges quite quickly compared to the more standard differentiable optimization that people would use in standard deep learning, right? So these different curves corresponds to the different differentiable optimization techniques, right? And the blue one is TextGrad. You can see that it actually converges to sort of a binder, and also relatively quickly, just after a few iterations. So we are excited about this TextGrad as one approach to try to help us provide a more flexible way to optimize these AI agents. Right? We've seen that you can use this to optimize agents' the ability to design molecules. In the paper, which I don't have time to go into now, we also show that you can use this TextGrad as a way to optimize, let's say, treatments, different radiation treatment plans for oncology patients, can improve coding abilities, and also improving like sort of scientific reasoning capabilities. Great. So I'll wrap up here, right? So I wanted to show these examples to illustrate, why we're quite excited about the potential of AI scientists as sort of our research partner, right? So through the virtual lab, we can see this platform where an AI professor working with different AI scientists with different expertise, they can work together and already solved some quite challenging and open research problems, like designing new nanobodies that bind well to COVID, which we've experimentally validated. Right. And this also motivates some new challenges about how do we optimize these more complex agentic workflows, which are not differentiable. And to overcome those challenges, we developed TextGrad as a non-differentiable optimization method that we can use to optimize these more complex, any of these agentic systems. So importantly, I want to acknowledge and thank my students and collaborators that led these different projects. So Kyle Swanson led to the Virtual Lab project together with our collaborators, John Pack's group at the CZ BioHub, right? And then TextGrad is led by a fantastic student, Murtyuk Sekunol, with a bunch of other students in my group. And I also want to thank our support. Great. Thank you very much.